So now hopefully you've had a chance to kind of work through some of these papers. Hopefully you're continuing to develop and generate your annotated bibliography. And the goal here is to find one of the papers that looks interesting to you. You've kind of dug into it a little more, and now you're going to present this one and another one. So you don't have to do these. You can change your next one if you're still kind of thinking like, I don't know if I want to look into this more or not. This is kind of that exploration stage for you to say, hey, this is something I'm interested in, or maybe not, and maybe in the next journal club I'll have something completely different, and that's totally fine. So this could be something that helps you develop your paper that you're going to develop by the end of the class, or maybe it's just exploring something that you think is interesting right now. The goal of this is, again, exploration and just also sharing the information with your peers and fellow classmates. So plan on about a 15-20 minute presentation of a journal that you find interesting. You should include in that journal, and I'll go through the rubric at the end. What are the aims and the goals of the hypothesis? What were they trying to do in the paper? Why were they trying to do it? Who cares? That's kind of a big, kind of zoomed out question. What is it? What is the point of it, and why are they doing it? Some of that stuff you've explained, maybe something similar in some of your earlier posts, what you're interested in, why you're looking at these things. But what were the aims and goals of the authors? And that should be a part of that. You should include a figure that you're going to present and how that fits into what they were doing and what they're looking at. And you also need to probably, whatever the figure is that you're presenting, you should also describe the technique they're using and how they go about collecting data to present and to generate that figure. So what techniques are they using? Is their data collection working? What tools or instruments are they using, and how do those things work generally? Again, this is a 20-minute presentation. We might not be able to do a full detailed analysis of a gas chromatography mass spectrometer, but you should be able to at least kind of provide a broad overview of that. And just assume that maybe your audience isn't an expert at that particular piece of equipment. You should go through just that one figure, that result, but then you can kind of zoom back out and provide a bigger picture. What do their overall findings mean from that, from the whole paper, not just the one figure that you showed us and walk through. And then any other aspects that you think that would be necessary to understand what they're doing, where they're going, what are the next steps, and stuff like that. How this should be organized, and I'll just kind of show you a brief kind of overview of some of the things to consider and think about. But one of the big things is this, when you generally do a presentation for a journal club, this is not a mystery novel. The chemistry and biology and biochemistry, those journals are thick enough and hard enough to figure out without trying to make it a kind of surprise ending. Just tell us what we're going to find out right away. That's part of the reason that a lot of these journals have abstracts. They're not trying to be mysterious, even though sometimes that content is very thick. They're trying to tell us right off the bat, what are all the big picture things that we need to know. And that's kind of what you should be thinking about too. We want to know what is their kind of punchline. We want to know it right away. So it's not a mystery novel. Don't try to wait until the end, like, did they solve the problem or not? Just say like, hey, they did do this, but they didn't do that. Just tell us right off the bat. Don't make people guess and wait to find what the conclusion is. It'll help as you're walking through it, and for the listener anyway. You should talk about their hypotheses and aims. Again, I mentioned that. Overall conclusions, what's the problem for the background, the model system, the techniques that are used. You need to at least present one figure, and this should be data that's collected. So tables should be last resort if there are no figures. Typically, if it's a research article in chemistry, there'll be some type of figure, not necessarily a table. So you can use a table to help clarify things or provide an overview, but you should have a figure. These again should be peer-reviewed research articles, not review articles. You can bring stuff in from review articles or pictures or figures if that's necessary, but these should be scientific journals with data. Bring it back and provide overall conclusions and conclusions you have, maybe where you want to go next with this, or what things you're looking for, curious about, and how this might lead you to your next paper that you're interested in. One thing to recognize and note are that these research articles are not a one-stop shop for understanding everything. And just recognize that the authors are assuming that there's some knowledge in the field. They don't have enough space. They're usually given only so many characters and space that they can use to put their information in there, and they can't write a whole entire review article about all of the background information. So usually the background information is pretty concise, and it will reference other things that you might want to look at. So think about that. You might need to use some other areas or reviews or other articles that they reference to help get caught up and figure out what you need to provide as some background information. Supplemental figures and papers, you might need to go into there and look at those a little more closely. And also be visual. Please don't include just pages and pages of text that you're reading off. That's pretty boring to listen to. So try to have some type of visual that helps to keep your peers engaged in what you're discussing. In these next few slides, I just want to show you maybe some examples of what I'm talking about in terms of what you might show or what things might be important to provide for a journal club kind of setup. So usually it's a good idea to just kind of get a screenshot of the PDF. Here I'm looking at a PNAS article, so the Proceedings for National Academy of Science. You can clearly see, oh, look, it's a research article. If it were a review article, it's a review. Some journals do this, some do not. But it's a nice way to say, oh, hey, I know for sure this is a research article. You can read out the title and look at any authors or where it's coming from. That's not super necessary, but if now is a good time to say, hey, this is what, looking at the abstract, they really wanted to look at this disease called spinal muscular atrophy, provide a few details about it. Don't spend a ton of time, but you can just kind of provide some of the overviews. And hey, they're looking at this activation of this kinase to see if it's involved in the disease progression. So that's what they're essentially studying, this enzyme. And then we'll kind of go into some of the figures that they looked at. So sometimes in journals, they'll have huge figures that have multiple sections, A through F, G through H. You don't have to present every single piece of information in a figure. So if you don't want to, you can just cut out. Let's say I want to look at A, B, and C, and I don't want to do D, E, and F, or G, H, I, and J are a whole different system and a different technique. So sometimes it's not going to be possible to kind of put all of these things in here and do all of the things that you might want to do. So you can break these things up. So this was one figure. I kind of broke it into two. Maybe you just want to talk about one kind of component of it. So on the left side is they're using immunocytochemistry, and on the right side they're using Western blot. They're looking at different things. So my recommendation is in 15 to 20 minutes, you're probably not going to be able to cover all of this in as much detail as you might want. So just one example here, let's say we're going to focus on the left side here with the immunocytochemistry. Here, I might have, before I talk about that, I say just before I go into this figure, this is how immunocytochemistry works. So they took stem cells, they plated them on plates. They used two different antibodies. The primary antibody was against, there were multiple different things. They looked at islet 1, TUJ1, OCT4, nestin. You might want to talk about what those are and why they're important. And then they did imaging on a microscope, and then you get these really cool fluorescent images. And this is the figure, and I'm going to walk through, what we saw, where we saw it, and then you might need to go into what the into the paper, say the reviewers thought that this was interesting, that islet 1 was expressed in these cells, or whatever it was. Maybe instead of immunocytochemistry, you wanted to talk about the Western blotting, and you say, hey, this is how Western blotting works. We have SDS, it covers all the proteins, we run on a gel, we use antibodies, and then we see a blot. And this is the data. They used these different types of antibodies, and here are the results, and this is what they found. So this is kind of the overview for not only the data, what you're presenting. You want to talk about it a lot more thoroughly than I did, obviously. This is just a quick discussion to kind of give you an idea, but you're going to want to walk through the protocols. So you might need some type of visual aid to help walk people through that and make it clear. And then you're going to show us the data and then walk through each panel of the data, what that finding was and why that matters to their overall goals that you mentioned earlier in the talk. Hopefully along the way, you're kind of tying this in with their goals and their aims, and that's kind of continually being discussed throughout the presentation. And then at the end, once you've discussed the figure at length, then you want to kind of go through thinking about the big picture. So what's next? So not only for the paper, so you want to, what are the overall findings for them? What are the next steps that they're going to do? What about for you as well? Are you going to continue looking in this subject? Was there something in that paper or in the discussion that led you to some other paper or some other thing that you might want to do? So start talking about not only the next steps for the research that you are exploring, but what about your next steps for this process of trying to understand more about some type of system? So hopefully that kind of provides a little bit of an overview of the type of things I'm looking for in your 15-20 minute presentation. The rubric is going to kind of look at some of the background. So did you provide enough background? Did you give some goals and aims? Do we know what the authors were trying to do? How well was the system, model, equipment discussed? Did you describe some of that in enough detail for the viewer to understand what you're doing and how those data were collected and presented in the figure that you're going to show? So the figure description, that's going to be part of this as well, is all of the information. And again, that relates back to how well you did on the discussion of the model system and the model. Did you go through all the axes and describe what those units are and the different antibodies, or whatever technique you used? Was the figure described in enough detail for someone to follow along and understand what happened? Overall organization, presentation, use some nice visual aids to help with kind of walking through the material, just overall preparedness to present, and then how well the topic overall was discussed and developed, and then time appropriate. So I'm looking for about a 15-20 minute video as well. So hopefully that provides some information about the overall goals of the video. Please, if you have more questions or still are not sure, send me an email. How I usually make my videos, I use a video kind of editing tool called Screencast-O-Matic, which I believe is a free download. You can generally make about 20 or 30 minute videos for free. I usually upload those to YouTube, so then I can kind of save memory space on my computer because the MP4s take up a lot of space. But you can do some simple editing and you can make some pretty good videos on there. So that's my recommendation. If you have questions about that, please let me know. But you have until next Tuesday to upload a video. And again, just keep in touch if there are questions, comments, or concerns you have about that. So thanks for watching.